Can we not?
If they remain like this.
Yeah, yes.
Are you able to fast forward?
Okay, thanks. Good morning, everybody. Welcome to the clinical rounds. I usually don't introduce the clinical round speakers, but today I'm introducing Pastor Tselios. He's a dear friend of mine and a collaborator. He saw me through the COVID days, and thank you so much for the collaboration. I've learned a lot about autoimmunity, the rheumatological, the clinical implications of autoimmunity, and we have had lots of discussions around how it can impact pulmonary complications. So today, when we were actually running short of a clinical rounds presenter, I reached out to him because I found this talk he gave at the allergy would also be fascinating, and maybe it will get some of our, lateral thinking processes started. So thank you for coming, and I'll leave it to Kostas. Just as a background, he did his MD in Greece. I'm not going to try to pronounce the name of his Greek university, but thereafter he did his fellowship, his PhD and his fellowship in University of Toronto, and then he got recruited here as faculty at the Division of Rheumatology. Okay, so I'll leave it to you.
Thank you so much, man. I don't mind actually, and I gladly accepted the invitation to give a talk to your rounds. You'll have to excuse me. This is my first in-person rounds in 5 years, actually. I spoke at the rheumatology rounds about 5 years ago before my hiring. He asked me to give a presentation about what kind of research I was doing in the University of Toronto. And since then, everything was via Zoom. They were surprised yesterday to learn from Victoria that they should come in person. So glad to see you all, and thank you for inviting me. The name of my university actually is Aristotle University. So I guess like most of you have heard about Aristotle. Like
I did not hear Aristotle.
Yes, guy 2,500 years like back. Okay, not a big deal. So to make it right, how can we do this? So I was so excited yesterday. After I realized that I was coming in person and seeing in person, that I got you baklava. Right, and you can say, right. Careful if you're allergic to nuts. Okay, lots of nuts. It's nice. Keep on for yourself. Right, so hello. Nice to see you. So, I thought to give this presentation about CAR T and not only T cells in autoimmune diseases. This is seemingly the next big thing in rheumatology, and I think that it will change lots of things in rheumatology in the coming years. CAR, chimeric antigen receptor. I will keep it simple. I see Dr. Bodis, actually Dr. Markey here, okay, but we'll keep it simple immunology-wise, right? Okay, this should change now, but I guess not. No? So this is the Sunday version of my name. They call me Costa every day, right? Or how my mother would call me when I was in trouble. Yeah. Are you changing or not? Right. Okay, the second slide is my. No. Okay, it's moving now. My disclosures, nothing to do with this talk. Okay, so the objectives, we'll try to review the biologic basis of CAR T-cell therapy, the current applications in systemic autoimmune rheumatic disease. I'm not talking about like the hematologic malignancies, and this is why and when and how this approach developed for hematologic malignancy. I'm good. It's working. We'll discuss the safety profile of CAR T-cell therapy. In future direction, we'll have like some thoughts about where we're going with this. So in rheumatology, immunologists know much more, right? For God's sake. But in rheumatology,
we say that B cells are the central drivers of autoimmunity, right? And you see, like you hear about autoimmune diseases, what pops in mind? Actually, like some autoantibodies that will give trouble. Is it only this? No, no way. But in some of our diseases, we have certain autoantibodies to blame. Have we ever shown a direct relationship between the autoantibodies and tissue damage? No, actually. Even for the well-known autoantibodies, like rheumatoid factors, for example, for rheumatoid arthritis, right? Do you know of any like model that's directly associates rheumatoid factors with the pathophysiology, the tissue damage? No. But in any case, this is moving on its own, eh? It's okay. So autoantibodies are coming from B cells, all right? We know that for good, when B cells not only produce the autoantibodies that we think they're related to the simple physiology, but also can present antigens, can like act as antigen-presenting cells, can secrete like a ton of cytokines or inflammatory cytokines or other cytokines and cause anyway trouble. So some of them may be autoreactive, we believe, in the case of systemic lupus erythematosus and other diseases. So in essence, we think that B cell, if we target B cells, we will achieve something for our patients. And what is the issue with B cells? It's not only one cell that will be generated in the bone marrow and will go on until death with the same phenotype, right? It will mature, it will differentiate, and you will see these like pro-B cell, pre-B cell, immature B cell, then they jump out of the marrow to the periphery, to the bloodstream. Mature B cells, memory, some of them plasmablasts, plasma cells, and then the plasma cells actually will start producing these antibodies, right? As the B cell is differentiating over time, the phenotype and the surface, all these molecules that make it distinct change. And you will see here that CD19 from the very, very first stages of differentiation, CD20 and the pre-B cell, and then they go on, at some point, they lose the CD20. And I'm talking about CD20 because that was the first marker that we targeted widely in autoimmunity, right? And we have drugs since 2004, 2005, if I'm not mistaken. So we have like a good grasp of how the B cells are differentiating over time and what are the markers that differentiate. So CAR T cell, did we need anything more in rheumatology? I was thinking like one could say we have 80 different monoclonal antibodies in rheumatology, for God's sake. We have destroyed every single health system in the world with the cost of monoclonals, right? And we still ask for more every day. And we run studies like every day for more monoclonals and more like fancy drugs that will cost like $1 million a year. Do we need them? I'll give you an example only. For renal disease in systemic lupus erythematosus, we call it lupus nephritis, right? Why lupus nephritis? Because with lupus nephritis, we have a very well set outcome. You have inflammation in the kidneys. If it doesn't go well, you will lose your kidneys. You will go to dialysis, renal transplantation, something, right? And these patients will have the worst outcomes overall, mortality, blah, blah, blah. Yeah. What happens? Why is it important? And when I'm talking about lupus nephritis, and we don't like pay more attention to diabetic nephropathy, for example, it's way more common. Lupus nephritis will happen in women 30 to 35 years of age. This is the age of onset on average, right? And if these patients do not do well by the age of 40, by 30, 35%, sometimes 40%, depending on the histologic class, will go on to dialysis. And then you will have to pay for these patients for the next 40 years. And patient on dialysis costs about $500,000 a year. Okay. So, and this young lady will spend the rest of her life on dialysis or praying to find a transplant. How we do now, or anyway for the last 20 years in lupus nephritis, right? That was the Aspirava study, mycophenolate versus cyclophosphamide. This is the standard of care. We call it now in lupus nephritis. All patients will take mycophenolate or cyclophosphamide, right? 8.6% complete renal response by 12 months. This is the outcome that will protect the kidneys at most. What we ask for a patient, or for any given patient with lupus nephritis, is a complete renal response by 12 months. Right, 8.6% of the patients at that time, Respirology was back in 2003, 2004. The rituximab trial in 2011, 30.6. Some other approach, multi-targeted approach, we call it, with tacrolimus and mycophenolate, 45.9. The BLISS-LN trial with the new, we had this for 10 years, but now we have it for lupus nephritis as well, the belimumab anti-BLISS trial, 47%. Aurora with calcineurin inhibitors, voclosporin, $100,000 a year for the voclosporin only, right? Let alone the standard of care. 41% success. And the University of Toronto Lupus Clinic, Dr. Tselios was doing his postdoc and supposedly like one of the best lupus centers in the world. This is what we do. 48.8% after 2003. That was the max. That was our best. So less than half of our patients achieve this outcome. So we do need something for lupus nephritis, maybe for other diseases that we don't have anything. I'll show you like a couple later. So, and some rheumatologists actually in Germany decided to act on this and see if this approach that was used in hematology oncology would be of value in rheumatology. What's happening? They will take the patient's blood. Yeah, they will isolate the T cells. They will get a chimeric antigen receptor gene in the genome with a virus vector, like lentivirus or adenovirus, and with something inactivated. Now they develop like new technologies with CRISPR. Yes, CRISPR. So they will avoid the viral stuff. They will insert the CAR. They will have these chimeric antigen receptor T cells, and then they will infuse this into the patients. Why do this? If you have a target, these guys, these T cells, these CAR T cells, will kill all the bad guys, or whatever guys you instructed them to kill. Right? Whatever you can imagine. And we had like third generation antigen receptor domain only. The second generation of CAR T cells with co-stimulator, like what they're trying to do, they're trying to expand the armamentarium of the CAR T cells. So not only to find the bad guys and kill them, actually, but have other actions on the immune system for better outcomes or for mitigation of toxicity. Right? What are the pros and cons of this approach? If you go to monoclonal antibodies, for example, here, we'll have like immediate availability, okay, like immediate figure of speech. But anyway, we can have it like now. I can have rituximab for a patient with vasculitis with EGPA. I guess with EGPA, you're more familiar to that term, right? Okay, we can have rituximab like in a few days. We can have like, okay, large manufacturing. We can have like as much as we like. Reduced risk with variability. It's well tolerated. Like we know these drugs relatively well, right? We have like clinical trials. We have observational studies. We have like large registries, national. We know how they work. What's happening with the costs? They have like a short half-life. Okay, rituximab goes for 6 months, and this like well respected. It should be okay, right? But other medications, other monoclonals have like a week or 2 weeks or a month, and this is it. And we have to do this again and again and again. And limited, of course, depletion depth. Why? Because it's difficult for these drugs to get to the tissue level or secondary lymphoid organs that we often see in autoimmune diseases. Right? They will kill maybe whatever like circulates in the bloodstream, but not deep down in the tissue. Like for example, we know that in lupus nephritis we have B cells, autoreactive B cells in the tissue, in the kidney, that keep causing trouble, and rituximab will never reach them. Now, for the CAR T cells, actually it's a living drug, we believe. You will infuse this to the patients, and then this will keep expanding and proliferating and doing a job for months, hopefully for life. Hopefully. Deep depletion, because they will go into the tissue and they will kill the bad guys. Okay, and this is very important. I'll give you some clinical examples. It's costly. Okay, and it's way more costly than the monoclonal antibodies. We're talking about $500,000 per patient. A lot, we'll see. I'll tell you like if it's a lot or not. We have some side effects. Okay, we'll discuss about this later. What was the issue with the B cells, since the CAR T cells, for the time being, are after B cells? We tried this rituximab in the LUNA study. We failed tragically. We tried it in non-renal lupus. We failed the Explorer trial. In the UK are still doing this, and they do rituximab with methylprednisolone pulses and not any oral steroids. Revolutionary for lupus. No oral steroids, my God. How is this working? It's working, and they showed like 50 people back in 2013. Now they had some failures, like showing complete or partial remission in almost 90% of the patients, regardless of the type of histologic class. So is it working or it's not working? What happened with the study? That was like a major issue in the lupus community. It didn't work. The official study was negative, right? Because of this, rituximab was never approved for lupus. We can use it like off the label sometimes or on a compassionate basis, or if we beg the manufacturer, right, something like that. But anyway, it wasn't labeled. They did a post hoc analysis actually of the clinical trial, of the LUNA trial, and they saw that if you have a complete B cell depletion, the CD20 B cells go down to 0, you'll have a 47% complete renal remission. If it's incomplete, 17%. And this happened in the study, actually. Some of the patients had an incomplete response, right? And we said that it works, the drug, when it works. When the drug will do what it's supposed to do, things will get better. When it does not, we don't expect anything. But we don't know who of the patients will respond. Now we have this obinutuzumab, a new monoclonal, right, anti-CD20 as well, but type 2 antibody, a much deeper depletion of the B cells. Again, the same target, but they modified it structurally. It's like a better depletion of the B cells. It's working better. This is the phase 2 trial. The phase 3 was positive as well. They submitted to the FDA. They haven't published the paper yet, but they submitted to the FDA. We hope that we will have this by the end of this year. Anyway, so we have some B cell approaches, right? So what is this issue? CAR T cell treatment. That was the first patient ever treated with CAR T in rheumatology, a lupus patient in Germany. Right, this is the patient, and the team of the doctors, 20-year-old patient, bad lupus with nephritis, with all the other stuff, and she has been treated with everything under the sun, or everything we have in for lupus patients. And you will see here, we have in lupus this anti-dsDNA titer, like we think that it's like a good marker of disease activity, like it goes up, activity goes up. Complements, this goes the other way around, right? An inverse correlation, this is activity goes down, this is activity goes up. So if this go like this thing, it's like a difficult situation, right? Proteinuria in the thousands, in the grams. I know some of you are not clinicians. If this goes on for a few years, the kidneys are lost. Right, done. And the B cells, because the patient was treated with belimumab, and rituximab as well. So, bad case, if this patient would not end well. The best possible scenario, kidney failure and dialysis or transplantation, right? So they go for coffee in the hospital's cafeteria with the hematologist, their friends, they talk to each other and they say, okay, you're doing CAR T, right? Thinking that maybe we can like have this to our patient. And they discontinue everything and continue only with steroids, depending on how the patient was feeling. They precondition with fludarabine and cyclophosphamide. Okay, we don't know much about this at day -3 and -5. And then they generate the CD19 CAR T cells, CD19 all the way from pre to plasmablasts, right? And they give this to the patient in that dose, like say, like 1 million per kilogram of body weight. What happened? These are the T cells, right? They went up by day 7. The B cells go down by day, it's like 26, right here. And then the anti-dsDNA antibodies in the thousands, that's a super high titer. After a few days after that, 0. The C3 goes up, gets to normal for the first time. And most importantly, the proteinuria goes down to 0 in a month and a half. Amazing, amazing. The patient did very well. And then since they have the know-how, they go to another 4 patients. And they give this to 4 patients, like 4 females. Most of our patients are females. SLEDAI-2K, this is like a measure of disease activity. In lupus, this is like moderate to severe disease activity. All had active kidney disease, and that was the issue, right? All were treated with whatever was available, plus the available biologics. They came up with this therapeutic protocol after the first 5 patients, right? And this is what I told you, okay, leukapheresis, they -10, then they fix the CAR T cells. Now we can have it. They say, like one manufacturer told me something about 2 days. I don't know like if it's true, but we can have it in 2 days, they say. The conditioning therapy with fludarabine and cyclophosphamide, then you give the CAR T cells. And then you keep the patient inside, inpatient, right? You are on their pillow, right? Looking out for trouble for 10 days. What happened with the CAR T cells and the B cell depletion for these like 5 patients? The CAR T cells were from 0.31 to 27.9, from 2.86 to 41.18, and so on, the expansion in 9 days. Okay, you give this to your patient, by 9 days, you're good. They will expand. And this is what happened, like if you want like a graph, right, the CAR T cells. What happened with the B cells? The B cells went all down, the CD19, right? We went after the CD19. So you expect 0, 19 in the patient's blood after a few days. White blood cells, okay, they will drop because of this. CD4, CD2, monocytes and neutrophils, everything will go like down, and then they will come up in a few days, okay? Usually in about 10 days. Clinical response, great. All of these patients, this is like a 2, it's nothing, most likely because of serology. Proteinuria down to 0 in 4 out of 5, like 4 had the significant proteinuria down to 0, like in 3 months. Complements up, okay, things are going well. dsDNA down, yes, yes, yes. Fatigue, I will not say much. Okay, we can't say much about like 5 patients. So of course, these patients had active disease. They're expected to have fatigue. What's happening with the B cells? Okay, we killed them, right? And we're out of trouble for life. What are these patients are going to do without like B cells? They will reconstitute at some point, and they will reconstitute not as memory B cells or activated B cells or plasmablasts, but as naive B cells. So new, not so bad or good guys, maybe, will come up after that approach, right? According to this. They recently published something like in 8 patients. They had another 3 patients from the States. All of them did well. Whatever we saw with the German patients, we saw with the American patients as well. Everyone responded very well. Now, we said about the CD19, right? Is it possible with plasmablasts, for example? The plasmablasts are the cells that secrete the autoantibodies actually are not bearing the CD19, so you cannot kill them. They bear this BCMA, B cell maturity antigen, right? And there was a group from China who thought of a dual targeting, actually, of both the CD19 and the BCMA. They developed this dual CAR T cells. And they were going after the memory B cells with the CD19 and the long-lived plasma cells, troublemakers, with the BCMA. They applied this to 12 patients with active lupus nephritis from active lupus from China. I think most of them had nephritis, similar therapeutic protocol. Right, they had some grade 1 cytokine release syndrome. I will talk about this, no neurotoxicity, some hematologic toxicity, but anyway, no significant issues safety-wise. Right, the patient did well, even with this like double target. And then they went only exactly from with patients, 2 patients with active lupus nephritis from China, similar therapeutic protocol, but slightly more CAR T cells, like 3 million. So, okay, lupus nephritis maybe, I don't know. We'll see. They achieved medication-free remission up to 46 months and only mild cytokine release syndrome. I heard about this recently. That's why I told you CAR cells, like not only T cells, the Chinese are developing NK cells, CAR NK cells for systemic lupus erythematosus. That was presented in the American College of Rheumatology in November. I was there. They have like 20 patients with active severe lupus, and they used NK cells, and they engineered the NK cells to go after the CD19 B cells. Why NK cells? I don't know, actually. When I asked the investigator, the primary investigator told me that they have the know-how with NK because they use it in China for anti-aging. Exactly. That was the reaction of the audience. Anyway, like different levels, different primary endpoints, all of this, and they do well, right? They only had like minor side effects. They did not do as well as the CAR T cell, like effectiveness-wise. Safety, okay. Reconstitution of B cells in 2 to 3 months, as seen previously. Why they develop this? Because they believe that we should have something off the shelf, something ready to give to the patient and not having all these procedures with like 10 days waiting or 3 days or 4 days, whatever the manufacturers can do to get it. Overall, as we speak, we have evidence in about like 30 patients, excluding China. Okay, from Germany, from the States, from other European countries, like 18 with lupus and 7 with systemic scleroderma, 5 with inflammatory myopathies. Median follow-up of 1 year now, so we have like some idea of what is happening and what to expect. The CAR T cells that you infuse in the patients stay there for about 47 days. The mean time of B-cell aplasia, we're talking about 3 months, and then they will start coming up again. Safety, okay, mild issues. Infections, mild, and some late neutropenia, like after like 3 months. They gave them like GCSF and everything went well. So this is the experience we have with this. I'll show you a few things about anti-synthetase syndrome. Anti-synthetase syndrome is a like an inflammatory, idiopathic inflammatory myositis. We don't call this the IIM anyway. Where idiopathic, you can say like immune-mediated or autoimmune, like most of the idiopathic stuff are nowadays autoimmune. So they had only 1 patient. It was published in 2023. And this patient was treated with everything like rituximab, again, rituximab, IVIGs, plenty of steroids, tons of steroids, cyclophosphamide, tacrolimus. And this is the CK. The CK is like a muscle enzyme, right, that goes up when you have muscle trouble. So you have like muscle damage, the CK goes up. And it's like a good measure of assessing disease activity. Up, 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 all the way. And then CAR T cells and down to normal, right? And you will see this here. By day 90, in 3 months, everything went down to normal. The antibodies, the titer of antibodies, anti-G1, this is the characteristic antibody for anti-synthetase syndrome, 1 of the 8 or 9 now, I think. This is what happened with the patient's MRI. All this fatty infiltration and edema went away in 3 months. Done, right? And because I'm talking to respirologists, I thought I could have this and see what's happening with the ILD. ILD is a major issue with myositis right? And this is our main trouble in these patients, the respiratory involvement. Even the respiratory involvement, much better after 3 months only. Long-term outcomes in 3 patients, right? After 12 months, the same CK went down. They have the manual muscle test where we can assess how the patient do clinically. The ACR total improvement score and extramuscular myositis is a systemic disease, right? We have like symptoms from other organ systems as well. Everything was much improved in these patients. Systemic scleroderma, another major trouble in rheumatology. We don't know what to do with patients with progressing, aggressive systemic scleroderma. Everything went well as well. Like 4 patients in the long term. This is like the modified Rodnan skin score is how firm, how affected the skin is. And even this went better, like showing that we have an element of inflammation in systemic scleroderma and not only fibrosis. We consider this to be fibrosis and irreversible damage. And it seems that we can do things for these people as well. Other applications, myasthenia gravis, I think like it's 1 patient or 2. Now multiple sclerosis, right, where you have some MOG producing B cells to go after. Sjogren's syndrome, this patient was had lymphoma and she was treated for lymphoma. Sjogren's syndrome, amazing response. IBD, dermatomyositis, adult onset still's disease, and many others, right? We have about 67 clinical trials now trying to recruit. I'm not so optimistic for this, but they try to recruit now. Can we aim at other targets? I told you, whatever you like, whatever you imagine, whatever like you dream of, you can go after, right? If you can find the funds to do this. This is like the card 19, what we were talking about. And this is the CAR cytotoxicity B cells that produce specific autoantibodies. So if you have, say, in pemphigus vulgaris, a very specific skin disease with very specific B cells that secrete this desmoglein-3 antibodies, you can go after them as well. Why? You don't need all the CD19 B cells to be killed. Not all of them are bad, right? Some of them may not give you trouble, so you may retain them. So we can be like more specific and go after like the very cells that would give us trouble. I don't know about this. If we can engineer CAR T cells with regulatory activity that can at the same time kill other cells through perforin and granzymes, suppress other functions through like the regulatory cytokines, IL-10, TGF-beta, IL-35, like even if they modulate other arms of the immune response for the patients. And this is what is being developed actually for preclinical models in multiple sclerosis and type 1 diabetes. Safety, a few things, and I'm done. Cytokine release syndrome. Why? Because you will be there. You will get in battle, right? Shooting and stuff like this, like cells are dying, cytokines are released. Yes, you expect this, right? Cytokine release syndrome, not so bad as the hematologist. Why? Because we don't have the bulk of the immune cells that need to be killed in rheumatology. We have a few bad guys, not like a clone that will get like everywhere, like acute lymphoblastic leukemia or multiple myeloma. So usually within the first week, okay, these are the cytokines that are thought to be involved. IL-6 and tofacitinib helps a lot. And tocilizumab, sorry, helps a lot. Very rare in systemic autoimmune rheumatic diseases, mild in most cases due to less B cell burden, right? Less B cells to kill. Neurotoxicity, what is this? This may be trouble. This may be trouble because it may present with aphasia, the patient cannot talk, or have some difficulty in talking, tremor, lethargy, seizures, even coma. Right? Not good. Why this is happening? Usually with the cytokine release syndrome, but may happen independently. They say something about IL-6 and MCP1 in the very first few days of the after the infusion. We haven't seen this in rheumatology, thankfully. We're very happy. Haven't seen this yet. They say up to 67% with acute lymphoblastic leukemia, up to 62% with lymphoma. I guess the cellular burden essence here. Other challenges, funding, right? A lot, an awful lot. Specialized facilities and stringent quality measures. We have this. We are very lucky to have this in Hamilton, and our colleagues up in Germany are doing this for many years now, and they have expertise in that, and they're doing very, very well. And patients who had no hope, like 10 years ago, now they're doing well. Contamination with viral vectors, not so much, and the issue is the allogenic, like off the shelf, ready-made CAR T cells. So, very promising results in systemic autoimmune diseases should be tested in RCTs, randomized controlled trials. This is 67 companies are trying to recruit patients for CAR T cell therapy. I don't know if they'll make it. What are the questions? Like, what will be the target? CD19, BCMA, double, other, maybe for other diseases, like if another cellular subpopulation is of more interest. The optimal dose, okay, usually like it's 1 million. If we can engineer other cells like the Chinese and go with after macrophages, I don't know, Dr. Bodes, if this would be like of interest to you. They ask about biomarkers to predict response to CAR T cell therapy, but everyone responded. Right, okay, like 30 patients all over the world, but everyone responded. Off-target effects, we don't know what is happening, like if we kill everything. And my major question would be like when in disease course and if this would change our approach for T cells and what I mean about it. If you go, where is it? Right, imagine you're 20 years old, right? You have a disease that will kill you. You don't respond to anything. Whatever Western medicine anyway has to offer. Right? And they tell you, you will give me $500,000, but you will forget about your disease. This patient is now 3 years drug-free remission. Drug-free remission is cure. We would never say in rheumatology. Cure. How much this patient will cost to the system actually, like for the rest 60 years of your life, 65 on average, I don't know what they have in Germany. If this is the case, naive B cells, right? In all, forget about memory, forget about plasmablasts, you will have new, young, strong, good guys to come over and take the place of the previously autoreactive B cells. So like a immune reset. Would you take it from day one of diagnosis? Like lots of questions, right, for this. But anyway, thank you very, very much for your attention. Yes, I'm done. Thank you. Thank you.
Thank you so much. Any questions from the in-person audience? Yes, yeah, Dr. Godish.
Oh my gosh, that was so exciting. Thank you so much for sharing. Is there, I guess I have a few questions. So
Yeah.
they'll have these naive T cells, but presumably they won't be able to differentiate into memory cells or plasma cells.
The T cells, the engineered T cells.
Yeah, the naive B cells, will they be just, like say that person gets vaccinated?
Right.
Will they be able to generate memory cells to that?
Yes. There are some data actually on vaccinations, the primary vaccinations like MMR and stuff like this. The titers were not affected after CAR T cell therapy. They did vaccinations with pneumococcal vaccines after that because the titers were like 0. They did other vaccinations, but the childhood vaccinations were there, present.
So my other question sort of related to that then is, over the course of decades, is there any evidence that these people will stop expressing CD19 on memory cells? Because we know that like it's not like in cancer, in myeloma, this happens frequently, and then
Right.
treatment because it's not biologically necessary for
Right.
cells to express.
We have data from the hematologists actually showing that the memory cells will come back. in hematology, right? Not hematology. We have some relapses in terms of new lymphomas, different type from what the CAR T cell therapy was applied, or leukemias that somehow that stem cell evaded or was differentiated, and after like 5 years or 10 years, it gave rise to the new cancer, right? We don't have this like length of data in rheumatology. It's only like about like this patient that it was first in like 3 years now.
So may I ask that follow-up question? So it also sounds to me like this is an incredible reset where the mechanisms of peripheral and central tolerance will sort of have a reset because the microenvironment is going to be altered by the treatment.
Right.
So has anyone looked at like T-brave cells, numbers and function pre and post treatment? Because it seems to me that there might be an add-on.
They were not affected. They were not affected. We checked like 25 high, 127 low. If I'm not mistaken, this is how they characterize it. And they didn't find any significant issue with that.
The numbers were different. You might expect a functionally in a different microenvironment, right? They might.
They didn't have any functional adaptation of difference. It was only a measurement of the and we don't have any evidence, at the tissue level, what's happening. Again, if they indeed uprooted the beat loss, it's very important.
Thank you so much for the presentation. That was true. I have a long list of questions here as I was generating ideas. So I guess what my question is I don't consider, and I know you know this well, I consider all the connective tissue diseases to be similar or the same. They're obviously very different.
Correct.
And I would argue that the relative contributions of inflammation versus fibrosis in those conditions is variable as well. And in respirology, we deal with a lot of myositis and scleroderma that sort of, by the time that it gets to us, it's already quite established fibrosis that's there. So I guess what my question is it naive to think that just targeting the B cells in diseases that are obviously multi-compartment as it relates to like the balance of inflammation and fibrosis? Like it was interesting you threw out your like the CAT scan. Like why did you put up that slide? Show me CAT scans.
I would never believe it.
But I would argue that was quite a bit of organizing pneumonia, maybe that was evolving, that it could have been like it was a small image, but
I would never believe it would happen myself.
So I guess what my question is like, do we think that this works well with lupus? Like, do we have like in these patients where they're treating the nephritis, did they do renal biopsies that showed that like the histopathology of the disease has changed? Not only that they're not putting out protein anymore. Like I was just curious as to like, is it just as simple as the B cells? Like, is it really that easy? Like,
I would expect like a totally different scenario, actually, that it would not only be B cells. It would be like a multitude of cells causing trouble. Right. Yeah. But I don't know where it went. Anyway, I would expect like a multitude of cells to cause trouble. Right. But is it possible that this patient was diagnosed early in the disease and some of the fibrosis appearance was inflammatory in nature?
Oh, certainly.
And then this patient.
We can stop it early on.
Yeah.
Well, that would be an argument for early initiation, right?
Yeah.
That's my issue. Like, and I was thinking, like, if you have this and it's going so well, okay, let's give it on day one. And have the patient like gone forever. Healed, cured. Like, can you imagine? Would you say that to your patients? With ILD, cure?
I never used the C word.
Right. Yeah. Me too. Me too. Like drug-free remission now for 3 years.
And so, in terms of the other question was the expansion of the actual CAR T cell pool.
Right.
So, is that because that you're dealing with a progenitor cell that you're it's not a transfection, but like that's being that the gene being inserted into is a progenitor cell is why you get that expansion within over time that you're seeing
I don't know. I don't know. They're biological. Like, I don't know what they call. They will expand, they will proliferate from day one. They hit the peak by day 9, and then they go away by day 47 on average.
Okay, so in terms of the duration of effect of these cells within the body is considered how long? Like it's 6 months, is that usually?
These cells will come back in 3 months. The naive T cells in 3 months, right?
Yeah.
But the effect of the disease was like for 1 year or almost 3 years I've been doing the first visit. Like no anti-TSA injectable for 3 years. No proteinuria for 3 years. No disactivity for 3 years. For scleroderma and myositis, we have 12 months. No anti-1 for 12 months. CBK.
Nathan, the way sometimes I look at it from the absolute immune software angle, as in 1 down to ground 0, how you might feel. If the presence of the autoantibodies, if they are not worked up, they keep on, even at very low titers, they may keep on doing their downstream activity, leading or contributing or promoting or facilitating the fibrotic and whichever other downstream processes that are going on. Right? So if you can take that source away, then chances of that having a long-term, in long-term, reducing the other effects, downstream effects goes down. This fibrosis is more active. You just don't know on what is going on. How much again the inflammation is causing the fibrosis leads because all the clinical symptoms are so difficult to manage.
But I think Nathan's question was, why are the T cells proliferating? Right?
I don't know. I was talking about this other visual one. That I do not know because I was not into it. I think there is they do a modification there.
Because they're effective memory T cells, which
In general, they proliferate, right? So they just that, and there was also a modification they did in one of the games, but I really did not understand it very well. So I just
I don't know exactly why. Like, they don't give any factors or something for them to expand.
it's just naturally
Okay,
being
these cells-
connected cells.
And they said that they only transfer the chemical receptor gene.
Yes, yeah. In
Right.
general,
And then
affected
the-
T cells should
yes.
proliferate. Affected T cell, then affected.
And then if the conditioning to have anything to do with it, like the cyclone just make everything else, I don't know.
Do you have any data on double negative T cells? Because I could see
No.
Nadia twitching.
No, I'm not aware of anything.
Nadia showed me a paper a few days back on
but according to the activated T cell phenotype, it's CD1 negative and CD11C positive, so that's the negative. So it seems that according
It
to
should
cell-
take
Right,
care of
right,
that.
right.
But once she showed me this paper, which was like associated vasculitis, so AAVs, and they were on Ritux.
Yeah.
And the non-response on Ritux in those patients were all associated with presence of double negative B cells, which
Oh,
means
I
they
see.
could not get it down.
Right, right,
So,
right.
and
right,
Ritux
right.
is CD20.
Yes, of
right?
course, of course, of course.
So that's what I was wondering.
Of course.
Hi, thank you very much for coming to talk to us about this morning. Where these cells that are put back in, do we know where they go in the body? Number one, and number two, are they also the effector cells for the biologic benefit that's enjoyed by the patient? And then the third question is something you mentioned way at the beginning, was that in lupus nephritis, you've evidence that there's some locally resident bad actors in the tissue that may not be accessed by rituximab therapy. And we, for decades in the lung, have been talking about the tissue determinants of disease. There's a microenvironment created within the lung tissue that determines disease outcome. And what you see in the blood or the bone marrow is of interest, but not necessarily determining. Correct. So where do these guys go? Are they the only way that some good happens from the intervention?
Right. And the matter is that we don't have evidence of biopsy post-treatment to see what's happened at the tissue level. And if you find any resident parts of that, it's only in the periphery. The only examples from the periphery. So they inject blood infusion. Yes, the peripheral blood. Okay. So they infuse it with the cells, and then they measure them from the bloodstream and not at the tissue. That would be of interest, yeah, to see what's happening there. And if there was, like, for example, we could have like pre and post biopsies to actually like see what were the differences. right?
But
We have
it would also
seen,
be very easy to label the cells and see, do they end up in the bone marrow, the lymph nodes, the diseased tissue,
with the radioisotope,
or something, yeah.
right? I'm not aware of any such
Or in
thing.
an animal model.
I think there is data on time.
Yeah. Going the,
The lymphatic
I would expect that they will go to the tissue.
lymphatic.
So you do, you don't.
I would expect that they will go into the tissues and they will eradicate all these like resident B cells. Like we have shown this in the price.
Yeah. Yeah, I wonder, it'd be interesting to know.
And they have like in preclinical model, yeah.
The primate, in the primate,
Yeah.
they showed it goes through the lymphatic, like it drains the lymphatic.
Lymph nodes.
Lymph nodes, yeah.
What about diseased tissue? Does the lung?
No, it was just
fibrotic lung
primate.
is a big place, it has a few intrapulmonary lymph nodes, you've got some lymph node tissue associated with mucosa, but very large regions of it are largely devoid of
Yes.
true lymphatic tissue.
Yes,
So
I
there's nothing there. So will these lads go, these effector cells go into the lung tissue?
That exactly is the point, and I was, my interest is always looking into localized learning
Yeah.
about it. So I have been, as soon as I saw it, that's the reason I've been looking into it, but so far, that's one of the things I found, this chance that I got to speak, because maybe we could have these discussions, because doing this better meeting was not helping.
You can go like,
Any other questions? Please, yeah, Dr. Henry.
What's this technology?
They have about 6 or 7 companies actually who are making this. They, I want to say something company, but okay. You might have like a major budget in this, and they run trials actually as we speak.
So when we are able, hopefully in the future, able to convince our hematology colleagues that this is
Right.
something that we want to be part of, we want to push for that. We have, who they have to have, it's not as though they have the technology that we can do on site, but it's something that's across the board.
They have
everyone's
it.
access
They
to.
have it now.
But they have to buy it from the company.
They, yes.
Like there's intellectual property there that has to have to be.
Yes,
That's why they're.
if we go outside the randomized control trial,
Yes.
right? Yes, we have to pay for it. We have to pay for it. I try to do this. I failed miserably.
And
To
Are there
convince
trials?
hematologist to give me one spot on.
And are there clinical trials in Canada ongoing with regard in the setting up?
As we speak, no. As we speak, no. There's a lot of pressure on us, because the issue is that they need centers that will have both rheumatology and hematology with expertise in target cell therapy. So we're talking about like Ontario, Toronto, and Hamilton. We're talking about Montreal, Vancouver, and Calgary. That's it.
That sounds pretty good.
Yeah.
Yes, but nobody wants to do this. Because multiple myeloma now has a trade indication for CAR-T, and we'll be sending patients with stem cell transplant to Buffalo. We can't accommodate it. It's impossible. And then if we ask for one research patient in the place of a clinically indicated patient, we may have trouble with higher, I don't know. I don't know, but really hematologists are not quite willing to do this now. Like, I would do whatever it takes to have like one patient for this.
I'm going to take one last question.
This is just a comment because I have experience working in the stem cell lab up at the University. And the CAR-T therapies, since they've been initiated, have been, it's a game changer for hematology patients. But the resources that go into them from both the clinical side and the lab side are just, it's extraordinary. So the cost of the cell production is $500,000, but the resources that go into the patient care and the lab resources, it's just, it is extraordinary. So I can see resistance from hematology because they have a lineup of patients that they want to treat with CAR-T therapies. And so to open up to something else, because it would still fall within the same lab structure, at least in the initial stages, the same clinical structure. So you're still looking for the same shared beds, right? And yeah.
It's a lot. It's a lot. It's a
yeah.
lot. I
fantastic,
agree
but
with
it's a
you.
lot.
My issue is this, if it's indeed curing,
Absolutely, yes. When you look at the long-term cost.
yeah,
Yeah, absolutely, but I think it's just beyond the conversation with hematology goes, you know, way beyond those resources. Hematology patients tend to be, pretty, I don't want to say, what's the word? They are sort of good for resources, right? Like cancer patients always generate.
it's
research funding.
different, yes, it's
yeah,
different,
They're good poster children for funding.
it's different, yeah.
When you go beyond that, it becomes less appealing to some, yeah.
So one thing I would say just to that, so we were doing a clinical trial, looking at endothelial progenitor cells in group 1 PH. And we went through the Dravinsky to try to initiate the trial for that. And for the exact what you mentioned, it just got shot down within like 30 seconds. Or like, that's really cool, that's really interesting. These were endothelial cells that were transfected with plasma that were secreting nitric oxide, which is perfect for the lung microenvironment, growth blood vessels, possible. But we were able to do it here. So because of for the freezes lab. So we have the ability to do the freezes in terms of the induction therapy. All that, and that would be difficult. Obviously, I think that would be a whole other realm. Because all we were doing was collecting the cells, then we were shipping them to Ottawa where they were being transfected, certified that they were sent back. We were administering it as outpatient as a therapy. So like, and from a phoresis lab perspective, like we have a unit here that we may be able to do that, but in terms of the lab technology and everything, that would be different. That has to be.
Well, and there is a debate whether it's actually, you know, cellular therapy versus pharmaceutical therapy. By the time the cells have been manipulated, it is almost, it is more like a drug, the way it's dosed, the way it's used. You know, so there's sort of that where you're looking at resources. It could be thought of as something different than cellular therapy proper.
Yeah.
I will have to stop the conversation. I'm glad we had a lovely discussion.
Thank you all.
Thank you, Konstantinos.
Good.